journey by pursuing PhD at the same institution. With a solid educational foundation, she earned her bachelor's degree from Government Kamalaraja PG College, followed by a master's in forensic science from Sri Vaishnav Vidyapit Vishwavidyalaya. Today, ma'am will take us on a deep dive into how forensic entomology is applied in real world investigations, from field protocols to data interpretation, as well as current challenges and future directions. Please join me in giving a warm virtual welcome to Assistant Professor Ms. Meghna Sharma. Ma'am, after the session, we will having a live Q&A. So please drop your questions in the chat box without hesitation. Over to you, ma'am.
Okay. Thank you so much, Priya. Thank you so much, Forensic team, to inviting me to deliver the lecture today. So today my topic is on the forensic entomology and how the insects which serve as the evidence or indicators to identify the post-mortem intervals of a particular person. So good morning all. Is I'm audible?
Yes, ma'am.
Okay, good morning. So let me start the PPT now and I'll share it. Is my PPT visible?
Yes, ma'am.
Okay. So let's start the topic. So my topic is forensic entomology using insects to estimate the time of a death or post-mortem interval. So first, let me tell you what is post-mortem intervals. Basically, a post-mortem intervals is the different stages occurs when a person is dead. There are different stages like a fresh stage and bloating stage, an active decay, an advanced decay, which occurs when a person is dead. These occurs because of the bacterial activity formed inside the body and the accumulation of insects that occurs on the dead bodies. Okay, so my PPT contains the PPT contains are is like first we will discuss about the introduction and historical background. We will discuss about the succession and life cycle. Then it is evidence analysis methods. Then there are some case studies and what will the future direction that can be implemented or can be uses in the forensic entomology cases. So first, let's just discuss about the introduction. Let me tell you the definition. Forensic entomology applies insect biology and ecology to legal investigation, primarily estimating postmortem intervals. Okay, so this definition is giving us the word that is forensic entomology. So entomology is basically a Greek word, entomon. Entomon means insects, and logy means we are segregating the two words. It means the study. So we are using the study of insects, that is entomology. So basically study of insects is entomology, and when we are using this study of entomology into the forensic world, then we can use this study of insects to implement in the forensic field and then to identify various post-mortem cases investigation purposes also. Basically prior era, a very before time I'm talking about, some experts says that forensic entomology is not the accurate word that should be used in the legal cases. So also they gave a word, some experts and some scientists suggested the word medico-criminal entomology. Medico-legal entomology that can be applied in the criminal cases. Now, but due to some advancements in the history, now we again back to this particular word that is forensic entomology. Now, what are the applications where we are using this forensic entomology? So, we use forensic entomology in the investigation, in the severe mass disaster cases, in the neglect cases of the old age peoples and the children's who are basically neglected by their own guardians due to the lack of care, and determining circumstances of death through insect evidences. Okay, so I hope my definition is clear to everyone how this entomology word came from, where from it is came from, and how with this entomology word is related to forensic, and where this forensic entomology is related to different cases. Now next come to the history. In historical background, we will discuss about how this forensic entomology word originated. So basically, first is originated from the field in 13th century with a first recorded case by Shang-Chi in 1235. What exactly what happened in China? The first case of entomology was recorded by Shang-Chi in 1235. Basically, Asangchi was a judge and a coroner. So he mentioned about this case in one of his book that's published around 1247, and the title was the book was Washing Away of Wrongs. Maybe you all are aware about that, or some of the people are aware about that. So what Shang-Chi mentioned in his book, that was name was Washing Away of Wrongs. That book was published in 1247, and the case was of 1235. So what he mentioned that in his village, the people used a tool that is a sickle. Sickle, you maybe know that the particular tool that we use to cut the crops or harvested crops or grass, that is curved like it is C like, जो हम हिंदी में बोलते हैं उसे हसिया, जैसे हम ग्रास या क्रॉप्स वगैरह कट करने के लिए यूज करते हैं. So basically, the people used it, and after using it, they used to clean it and put it under the sun to make it dry. So what he observed that some of the villagers, out of some villager, there was one person who used the sickle tool outside the sun to make it dry. That was lying out, but despite it is clean, there can he see the accumulation of different flies. That different flies indicate that maybe some of the blood spots were present on that sickle cell, or sorry, in that sickle tool, which gave the rise of like attraction of the flies. So by this, he considered that yes, some blood spots was present there, and he found that particular person guilty. So the first case of forensic entomology was came from there. Now the next we move to that is the modern forensic entomology developed to the work of Magnin in 1894. So, Magnin in 1894 was a military doctor, and he studied on the insect succession. Insect succession means it is a time-dependent sequence of insects of different species which form a colonies on decomposed body. If I tell it you clear, so insect succession is a, I can say, life cycle of a particular different species. Okay, like different, there are many multiple of different species, and different species have different types of life cycles also, and they are dependent on the environmental condition and on the time as well. And these species complete their life cycles on the decomposed bodies. Okay, so insect succession is a time dependent process or a sequence of insects of different multiple species that particularly colonize or grow on the decomposed body only. So in 1894, the Magnin, who was a military doctor, he studied about the insect succession on the decomposed body, and after studies this insect succession, he published the book that is Fauna of Corpse. Okay. Now, next will be the discipline gained scientific credibility through a systematic research in 20th century with a landmark case like the Ruxton murders. Maybe you all heard about the Ruxton murders. It is also known as jigsaw murders. In 1935, it is a very known case. What happened in that? Dr. Burke Ruxton was a very famous doctor in the UK. So he was having some doubt on his wife. So he murdered his wife, that is Isabel Ruxton, and her maid as well. And he dismembered both the bodies and threw it somewhere. Now, after some days, these dismembered bodies were found by the police, and they take it out, and they found that there was a larva of the blowflies. Clear? There was a larva of the blowflies on the dismembered bodies parts of the Miss Rexton and her maid. And when they calculated by the help of the entomologist that what particular the time cycle or particular the time period when the flies lay their eggs and the maggots found on that, they estimated that it is 12 to 14 days old. So this case was happened 12 to 14 days old and the body was dismembered and thrown out somewhere in the UK. And this is how the entomology field came into the forensic field and the mix up called the forensic entomology. Clear? Now, the next is insect succession. As I already told you all that what is insect succession. Insect succession, I told you that is a completely time-dependent process of different species of insects and they complete their colonization or a life cycle on a decomposed body. So, why it happens? Like insect succession hota क्यों है? So it happens due to the various reasons, due to the availability of the food, due to the types of tissue present on the body, and smell emitted by the body and environmental condition. So I told you that there are some species of the insect that complete their life cycles on the decomposed body. Why? Because some insect species complete require the
Excuse me,
food
ma'am.
that is flesh. Yes.
Ma'am, can you please zoom in the screen?
Okay. It is okay now? Hello. Is it okay?
मैम फुल स्लाइड करिए.
Full slide. Actually, my system lag after I doing this.
ओके मैम.
That's why I'm using it. Let me try. Is it? I did it from my side.
No, actually, it's the
Oh,
same. It's
it's
fine,
the
ma'am. You
same.
can continue.
Okay. So, yes, I was on the topic why insect succession happens. So this insect succession happened because some of the larvae and the maggots of the insects require the human body as their food. Also, the smell that is coming out from the human body that is happening due to the decomposition by the bacteria that attract some insects to perform their life cycles and mating process on that particular body. And also, I told that the human body serves as a food for some insects and their larval growth. So it serves as a nutrient things also for the insects, whether it is a dry tissue or whether this is a wet tissue. Okay. Now there are the different stages of insect succession, that is first stage, then bloat stage, and that is decay stage. तो फर्स्ट स्टेज इज फ्रेश स्टेज. पहले स्टेज जो होती है वो होती है आपकी फ्रेश स्टेज. फ्रेश स्टेज कब होती है? द बेसिकली बॉडी इज डेड एंड इट इज अप टू स्टार्ट डिकम्पोजिशन एंड द डेज अबाउट ऑफ डेथ इज 0 to 3 days. ठीक है, मरे हुए इंसान को 3 दिन हो चुके हैं. क्लियर? So, who arrived first? The blowflies. The blowflies that are basically belong to a family of Californidae. So they arrive within a minutes to hours. Okay. These blowflies are the first flies. Consider it. These blowflies are the first flies belongs to the family of Californidae. Comes to the decomposed body or a dead body. That when it is in a fresh stage between 0 to 3 days. It starts laying begins immediately. Okay, the egg lay by them start immediately. And if I tell you what is the average egg that is laid by the blowflies, it is around 150 to 200 eggs. A single blowfly can give up to 150 to 200 egg on the human body. And these eggs are laid into the open surroundings or open cavity of the decomposed body. Now, if we talk about the what are the open cavities of the decomposed body or what are the open cavities of a dead person, then we can say it is the body which are the body have different open cavities, like it can be your eyes, it can be your mouth, it can be your nasal cavity, it can be your private parts, or if a person have any open wound, okay, then also these blowflies can lay their egg in that particular area, and this is also a very good sign to identify that yes, the injury was there particularly. ठीक है, वैसे तो आपकी blow flies आपके different different oral cavities में एक ले करेंगे, that I already told you, that eyes because of the moisture present there, the nasal cavity have the moisture, the mouth have moistures, and the different private parts of the human body. But if a body have open wound injury, then also the blow flies lay their eggs there. Okay. The bacterial action start decomposition the body and temperature dependent colonization. This whole process is a temperature dependent. Okay. Now the second stage is bloat stage. Bloat stage happens when the days about reaches to 1 to 6 days. Okay. Now why it is bloat stage? Okay. Just tell me if I'm going very fast or if my voice is not clear. So bloat stage is the stage when the body start bloating. Bloating means gas form hona. Why it is forming? See, already it is mentioned there that bacteria action start decomposition. So when the bacteria starts proliferation or action, see, like how your blowflies are laying eggs and their colonization is also forming. Same like that also, the bacteria who are decomposing the body from inside, they are also forming the colonies. They are also proliferating. So when the bacteria are proliferating in large number of a cycle, so they are releasing a gas. Okay? They are releasing the gas, and that gas formed by them is hydrogen sulfide gas. Hydrogen sulfide gas is formed due to the bacteria that are proliferating inside the human body and responsible for the decomposition. That hydrogen sulfide gas is like inflammable also, and बहुत ही pungent smell type का आता है उसमें से. That gas is hydrogen sulfide gas is responsible for the bloating of a particular body. Okay, these bacteria which are proliferating inside the bodies are anaerobic bacterial. Okay, anaerobic means that bacteria does not require oxygen to proliferate. Okay, so this anaerobic bacterial gas production is nothing but it is hydrogen gas, hydrogen sulfide gas that is responsible for the bloating of the human body. This as a series of decomposition is performing, the tissues also start to liquefy. Liquefy मतलब गलना start हो जाते हैं organs का अंदर से. Then there is a massive larval feedings also, and peak insect activity start at that particular phase. Also, I want to tell you when the bloating stages reach, then another species of the insect that is Sarcophagidae, that is flesh fly. और वी कैन से हाउस फ्लाई ऑल्सो दैट मस्कीडे जो नॉर्मल हमारे हाउस फ्लावर्स में होती है. सो टू फैमिलीज, टू डिफरेंट स्पीसीज फैमिली, दैट इज सार्कोफेजिडेई, दैट इज फ्लैश फ्लाई, एंड मस्कीडे दैट इज योर house fly starts to arrive when the bloating stage reaches of the dead body. And also I can say some beetles also started to coming to the bloat stage, because this bloat stage attracts different species, your sarcophagidae, your muscidae, and beetles also. This sarcophagidae insects I want to tell you, as I tell you, the blowfly usually lay their eggs on the dead bodies. Sarcophagidae family species are different to blowfly insects. They are oviviviparous. Oviviviparous means Sarcophagidae basically lay the eggs that are about to hatch or which are already hatched. So they are different from the blowflies. That's why they are different from the blowflies. Now, next move to the decay stage. This decay stage is divided into the two categories. One is active decay and one is advanced decay. What happens in decay stages? Tissue mass reduction means the flesh of the body start reducing, means the wet flesh that is present on the decomposed body start to decrease in number. Beetle colonization increases. Beetles, who are the beetles? Beetles are basically the predators who for whom these insects or these maggots or the blowflies eggs and the sarcophagidae eggs or maggots serves as a food. Okay. Then predator insects arrive. Who are these predators? Predator insects like your beetles can also be. Then your ants could also be. Your spiders could also be. Different types of insects can also be arrive at that time. Here it is mentioned diphthera larva migrate. What is, can anyone tell me what is diphthera category? What is, why basically this category is diphthera? Okay, so basically diphthera species are those who have two wings. That's why it is known as diphthera, means two, and two wings. Okay. So these blowflies that arrives early in the fresh stage are belongs to the diphthera family. So when these predators or these beetles comes on the body, they start feeding on blowflies eggs and blowflies larvae. That's why it is mentioned that diphthera large family starts to migrating because beetles start to feeding on them. As I also mentioned, that decay stage have two different categories. First is active stage and second is advanced stage. Active stage is basically between 5 to 11 days. It takes 5 to 11 days for active decay of the body, and it takes 10 to 25 days for advanced decay of the body. When it reaches to active decay, the body start giving a fall odor smell. That is forming due to the hydrogen sulfide gas formation. That hydrogen sulfide gas giving a fall odor smell. That fall odor smell attracts the predatory beetles that comes on the body, and they start feeding on the maggots of the blowflies, which start giving the migration of the diphthera larva. Second is your advanced stage that forms after the 10 to 25 days. The remaining flesh that is left over on the decomposed body, it's about to dry. Okay, maggots that is formed on the body, maybe some maggots that are formed on the body of blowflies or of the Sarcophagidae family, they start to migrate into the into the soil that is nearby the body or the cloths of the body or the nearby leaves or something areas to form into the pupa form. Okay, to convert that when the maggots gone into the pupa form, then they can be converted into the adult stage. There are life cycle I will tell you further on. So, in what happened in advanced stage, the maggots that are forming after the egg stage of the blowflies, they start going or invading themselves into the soil and into the clothes of the decomposed body or the body, and may transfer them into the pupa stages. Now, the decay stage attracts the Dermistidae family. Dermistidae family is the beetles family. They are the predators animals or predator insects that feed on the blowflies. Okay. Next is the decomposition timeline. So I already told you that first is active decay, second is advanced decay, and after the advanced decay comes the dry remains. Okay, so what happens in active decay? Intense bacterial activity and tissue liquefaction with maximum insect colonization and feeding activity occurring during this phase. I already told you that the insect in bacterial activity start happening in the fresh stage only, due to that the hydrogen sulfide gas is formed and the body is bloated and the liquefaction of the body starts. And when the body liquefies, the insects, maggots, and feeds onto that particular tissues, like white tissues. Next is advanced decay. Reduced tissue mass, that is flesh, with specialized scavengers insects that are your beetles, replacing primary colonizations in the successor pattern. So that Sarcophagidae and Dermistidae family members arrive and receive.
Excuse me, ma'am.
Yes,
Sorry for
slide
the
is not
interruption,
visible
but slide is not moving here.
now.
Yes, ma'am.
Okay. So next I was talking about advanced decay. Advanced decay means जिसमें आपकी बॉडी का फ्लैश का लेवल या अमाउंट जो है कम हो जाता है, एंड दैट इज डर्मिस्टेडी फैमिली जो होती है, आई टोल्ड यू दैट इज बीटल फैमिली. डर्मिस्टेडी फैमिली बेसिकली आपकी प्रेडेटर्स फैमिली होती है, जो कि आपकी ब्लो फ्लाइज पे या फिर आपकी फ्लैश फ्लाइज पे या मस्कीडे पे हंट करती है. दैट्स व्हाई दे आर प्रेडेटर्स. ओके. So they replace to them. Okay. Then the last come the dry remains. What remains and the dry remains? So whatever the tissue of the body that will become dry, like your bone, your hair, your cartilage, remain with the specialized dermestid beetles completing the decomposition process. Okay. तो जो भी आपकी बॉडी में जो हेयर बचता है, बोन बचता है, ये सारे आपके जो डोमेस्टिक बीटल्स होते हैं. डोमेस्टिक बीटल्स कैन बी योर आंट्स हो गए, और इट कैन बी योर कारपेट बीटल्स हो गए. ठीक है. It can be your like we can say insects हो गए. ठीक है. So these are all the insects that help to or completing decomposition of the बॉडी, ठीक है। जो ड्राई रिमेन्स होते हैं, जैसे कि ड्राई रिमेन्स पे बॉडी में क्या क्या बचता है, जैसे कि आपका हेयर्स बच रहा है, आपके क्लॉथ्स बच रहे हैं। तो जो आपके डर्मिस्टिडेई फैमिली होते हैं वो बेसिकली फीड करते हैं केराटिनिन कंपोनेंट पे। एंड यू ऑल नो वेयर द केराटिनिन इज प्रेजेंट, इट इज प्रेजेंट इन द हेयर्स, ओके। सो जो आपके डर्मिस्टिड बीटल्स होते हैं वो आपके मैगट्स पे तो फीड करते ही हैं, साथ साथ जो आपके हेयर्स हो गए उस पे भी फीड करते हैं, और जो क्लॉथ जो अगर किसी बॉडी पे रह गया तो उसके ऊपर भी फीड करते हैं। नाउ नेक्स्ट मूव टू द इंसेक्ट लाइफ साइकिल। ओके, सो in blowflies or any species of the insects have four different level of stages during their complete metamorphosis life cycle or complete metamorphosis life cycle. First stage is your egg stage. As I already told you that a female blowflies can lay up to 150 to 200 eggs on a decomposed body. ठीक है? When you see how these eggs look like, they look like a rice grain. चावल देखें आप लोगों ने? If you see the pictures or if you see somewhere else that how these eggs look like of the blowfly, they completely look like a rice grain. ठीक है? एक चावल के दाने की तरह दिखते हैं. It takes about 8 to 24 hours to hatch them. Around 8 to 24 hours it takes for hatching purpose. Once these eggs are hatched, it comes to the larval stage. Your larval stage or maggot stage, it is same. Whether you can say it larval stage or you can say it a maggot stage. This egg stage reaches to larval stage after completing the 8 to 24 hours. This larval stage complete three instars with rapid growth and feeding activity. What is this three instars? These three instars are basically dependent on the shape of the larva, how it is growing. So this three instars is first instar, second instar and third instar. First instar जो होते हैं आपके उनकी लेंथ अराउंड 2 mm तक होती है। अब वो बेसिकली फीड करते हैं लिक्विड टिश्यूज पे, ओके। द फर्स्ट instar length is about 2 mm and they basically feeds on liquid tissues. Now comes to second instar. When they reaches to second instar, their activity starts more and they start penetrating the tissues. They start penetrating the soft tissues and feed on the inside tissues. Okay. Now reaches to the third instar. Third instar reaches to its maximum length, that is around 10 to 20 mm. And they start growing away from the body to प्यूपेट, ओके। जब आपका लार्वल स्टेज की थर्ड स्टेज आ जाती है जहां पे आपका लार्वा कंप्लीटली डेवलप हो चुका है एंड रीचेस टू 10 to 20 mm stage, at that time they become so much active that they start proliferating outside the body and roam around and go here and there beneath the body or beneath the soil that is lying around the human body or inside the claws, whatever is space is present around the body. And why they go? They go to complete their pupal stage. That is their third stage. What is pupal stage? A non-feeding development stage in protected microenvironments. Okay. Pupal stage में जो maggots हैं, they bury in soil beneath the body to pupate. Okay. To pupate means to to form their body into a particular morphological character. Okay. In the larva, it is a single structure. We cannot morphologically differentiate it easily. But when a larva reaches to pupal stage, in that pupal stage, it is a non-feeding stage where it do not require any nutrient or anything or any food. This larva complete themselves into a proper body formation that we can easily differentiate according to its morphological structures like their head, their thorax, their wings. Okay, so those pupal stage is very crucial and it helpful for a larva to develop into a proper body formation. It develops into a head, it develops into a thorax, at different wings, antennas, and different more so on things. So, how many did this pupal stage takes? Again, it pupal stage takes 6 to 14 days for a complete development to a adult stage. Once a complete development of a adult stage form, then this adult stage is a reproductive adults. They can again start their life cycle by mating and forming their own life cycle or insect succession. Okay. So Now the thing is how much time this complete life cycle takes. That complete life cycle takes around 14 to 21 days to complete this process. मतलब आपके एग स्टेज से आपकी एडल्ट स्टेज तक फॉर्म होने में एक एग से एक एडल्ट स्टेज तक किस तरीके से एक इंसेक्ट फॉर्म हो रहा है, इसके लिए आपको कंप्लीट 14 से 21 डेज लगते हैं. क्लियर? नाउ here are the some basic key forensic species that are responsible for the insect succession. First is your Calliphoridae. ठीक है, these are the primary colonizers arriving within hours of death. What are the hours? That is around 0 to 3 days. Calliphoridae से जो members belong करते हैं, that are your Lucilia sericata, that is green bottle fly, Chrysoma megacephala, that is oriental latrine fly, and Calliphora vaccinia, blue bottle fly. This green bottle fly and blue bottle fly are the basic glowflies that can be easily first see on the decomposed body. Okay, these are the first scavengers. Second is the Sacrophagidae. I told you about the Sacrophagidae, how it is different from the Calliphoridae. Sarcophagidae is a oviviviparous category that do not lay eggs directly on the decomposed body. Instead of this, they lay the that is hatching eggs or the eggs that are already hatched into the larva onto the decomposed body. Category of these are Sarcophaga tibialis, flesh fly. They larviparous reproduction patterns. Larviparous means they already laid hatching eggs that is converted into the larva or maggots. They are very temperature sensitive development. Second is your Dermistidae. Dermistidae family, I already told you, is a predator family that feeds on Calliphoridae and Sarcophagidae. They are dermistes beetles that are predator beetles. They are keratin feeding specialists. I already told you what is keratin feeding specialist means. They basically feeds on the dry remains of the decomposed body, such as your hairs or maybe your clothes. They feeds on bone and cartilages as well. Now the next is evidence collection. How basically the evidence is collected in the cases of insects. So what we have to do is that first the field or scene documentation is done. Photograph insect activity patterns in environmental condition, temperature recordings. Specimen processing is live specimens, preserved specimens, developmental stage, species identification, analysis, microscopy, measurement, so on. So let me tell you in the short things. What we have to do. First, we have to documentize or capture the particular body of that person. So we have to do it by using the photography, by capturing the photos and videos of that body and also the nearby surroundings like the soil, flora, fungus, whatever different things are present around the body, we have to capture them as well. Okay. Then we start capturing the insects that is present around the body, around the cell, soil, around the soil. Okay. So what we have to do कि हमें एक साथ सारे इंसेक्ट्स को कैप्चर या प्रिजर्व नहीं करना है, हमें सेपरेट सेपरेट प्रिजर्वेशन करना है. See, if we are seeing any types of stage of the insect like egg or larva stage or pupa stage or adult stage in any of the cavity, like अगर आपको बॉडी की किसी भी ओपन कैविटी में से कोई भी स्टेज आपको मिल रही है अगर स्टेज इंसेक्ट की, then you have to keep it separate. Like if you're getting it from nasal cavity, you have to preserve that category separate. If you are getting it from the ओपन वुंड यू हैव टू प्रिजर्व दैट कैटेगरी सेपरेट. आपको एक साथ सारे कैटेगरीज को कंबाइंड एविडेंस में कलेक्ट नहीं करना है, प्रिजर्व नहीं करना है. ठीक है. सो हाउ दिस डिफरेंट कैटेगरीज आर प्रिजर्व्ड. So when we are collecting the eggs, like when we are collecting the egg, we have to smoothly remove the eggs by using a brush and then we have to preserve it. How they are preserved? If we are collecting the egg stage of a particular insect, then we have to preserve in the two different category. First category में क्या करते हैं, जो insects होते हैं, sorry, जो egg होते हैं, we make them die by boiling, by preserving in than the 70 to 75 to 90% alcohol. Okay. जब हम एग को प्रिजर्व करते हैं, इंसेक्ट्स के एग को प्रिजर्व कर रहे हैं, चाहे हम सॉइल से प्रिजर्व कर रहे हैं या हम ह्यूमन की किसी भी ओवल ओपन कैविटी से प्रिजर्व कर रहे हैं, तो हम एग्स को दो कैटेगरी में प्रिजर्व करते हैं। फर्स्ट कैटेगरी होती है जिसमें हम 75 to 90% इथाइल अल्कोहल यूज करते हैं, जिसमें हम एग्स को रखेंगे। ओके। सेकेंड कैटेगरी होती है हम एज इट इज एग्स को प्रिजर्व करेंगे। दोनों कैटेगरी हमें फर्क ये देखना है कि जो हम 75 to 90% कैटेगरी में एग्स को रख रहे हैं, तो हमें उस टाइम पर टेम्परेचर को नोट डाउन करना है और एनवायरमेंटल कंडीशन को नोट डाउन करना है कि पर्टिकुलर एनवायरमेंटल कंडीशंस और टेम्परेचर कंडीशंस पे आपकी ये स्टेज फॉर्म हुई थी। ठीक है। नाउ द eggs that are preserved inside the 75 to 90% alcohol, now they will not be active to move on further process। ठीक है, वो वैसे के वैसे ही रहेंगे। और जो सेकंड कैटेगरी जिसमें आपने एग्स को एज इट इज प्रिजर्व किया था, उसे उसकी एनवायरमेंटल कंडीशंस के साथ प्रिजर्व करना है। मतलब अगर आपने एग्स को सॉइल से कलेक्ट किया है, तो आप थोड़े बहुत सॉइल पार्टिकल्स लेके उस कलेक्शन में रख सकते हैं। तो फर्दर ऑन नोटिफाई कि अगर आप नेक्स्ट स्टेज में वो किस तरीके से मॉडिफाई होंगे, क्या वो एग्स किसी और इंसेक्ट से बिलोंग्स तो नहीं कर रहे हैं, दैट इज वाई। Now next when we move to the larval stages, larval stages में क्या करना है, जिस तरीके से हमने eggs को प्रिजर्व किया था टू डिफरेंट कैटेगरीज में, सिमिलरली टू दैट हम लार्वा को भी दो कैटेगरी में प्रिजर्व करेंगे। फर्स्ट कैटेगरी टू किल देम एंड सेकंड कैटेगरी कीप इट एज इट इज। ओके। फर्स्ट कैटेगरी जो होती है जिसमें हम लार्वास को किल करेंगे बाय यूजिंग द बॉइलिंग वाटर। ओके। फर्स्ट कैटेगरी में हम लावा को किल करेंगे बाय यूजिंग द वार्म वाटर और हॉट वाटर, हम उन्हें बॉइल करेंगे और एज इट इज पैक कर देंगे ताकि वो अब फर्दर मॉडिफाई नहीं हो पाए। सेकेंड कैटेगरी जो रहेगी, हम एज इट इज लार्वा को प्रिजर्व करेंगे उसके एनवायरमेंटल कंडीशन या उसके सराउंडिंग कंडीशंस के साथ, टू सी इन विच नेक्स्ट कंडीशन दे विल बी फॉर्मिंग। एंड द थर्ड कैटेगरी इज टू द प्यूपा स्टेज और हैचिंग स्टेज। प्यूपा स्टेज को हम एज इट इज कवर कंटेन करेंगे। क्लियर। हम उसे हीट या किल नहीं करेंगे, हम उसे एज इट इज उसकी सराउंडिंग्स के साथ प्रिजर्व करेंगे। एंड द लास्ट स्टेज इज ऑफ द इंसेक्ट स्टेज और द एडल्ट स्टेज। एडल्ट स्टेज मतलब कि जिस तरीके से इंसेक्ट्स हैं उन्हें उसी तरीके से कलेक्ट किया जाएगा। ओके। सो हम इतने सारे स्टेजेस में अगर एग्स को, लार्वा को और प्यूपा को, इंसेक्ट्स को कलेक्ट कर रहे हैं, तो उसका बेसिक रीजन क्या है? बेसिक ये रीजन ये है कि वी वांट टू आइडेंटिफाई वेदर दीज कॉलोनीज और दिस स्पीसीज बिलोंग टू सेम इंसेक्ट्स, सेम फैमिली और नॉट, या फिर बॉडी का रिलोकेशन तो नहीं किया गया है। रिलोकेशन मतलब कि बॉडी पहले कहीं और थी, वहां के इंसेक्ट्स ये दूसरे पर्टिकुलर ज्योग्राफिकल कंडीशन में ग्रो कर रहे हैं, या फिर उन्हें ग्रो करने लायक वो कंडीशन मिलेगी नहीं, तो वो एज इट इज इसी कंडीशन में सस्टेन रह गई। क्लियर? So how we can, this is the basic step, like how the evidence collection to be done in the entomology cases and why it is needed. Now move to the analysis methods, how this evidence are analyzed. So they are differently analyzed into the morphological cases and the analysis and molecular analysis. By the name itself, it is very clear what are the morphological analysis. Morphological analysis are the traditional species identification, use detailed microscopic examination of larva and adult morphological features, including head capsules with spicular patterns, cuticular structures for accurate taxonomic determination and forensic investigation. Basically, what happened in this morphological analysis is the external features analysis of the insects by which we can identify that yes, it belongs to a particular फैमिली। मतलब कि उनके हेड के स्ट्रक्चर्स को देख के, उनके विंग्स के काउंट्स को देख के, उनके विंग्स के फॉर्मेशन को देख के, उनके एंटीनास को देख के, उनके बॉडीकुलर स्ट्रक्चर्स को देख के, बॉडी पे जो क्यूटिकल्स है उसके पैटर्न्स को एनालाइज करके हम ये आइडेंटिफाई कर सकते हैं कि यस, इट बिलोंग्स टू द पर्टिकुलर फैमिली। नाउ if the evidence data collected in the insect cases are not proper, और मतलब कि बहुत मर्ज है या प्रॉपर इंसेक्ट्स का कलेक्शन नहीं किया गया है, या इंसेक्ट के कलेक्शन के टाइम पे आपके एविडेंस में कुछ वी कैन से चेंजेस हो गए हैं, या वो प्रॉपरली कलेक्ट नहीं, कुछ भी चेंजेस हो गए हैं, देन वी रीचेस टू द मॉलिक्यूलर एनालिसिस। मतलब कि अगर इंसेक्ट्स का कलेक्ट करते वक्त, अगर उसको मॉर्फोलॉजिकल एनालाइज करते वक्त अगर में हमें कोई प्रॉब्लम हो रही है, हम उसे मॉर्फोलॉजिकली डिफरेंशिएट नहीं कर पा रहे हैं, सेग्रीगेट नहीं कर पा रहे हैं, आइडेंटिफाई नहीं कर पा रहे हैं, then we will reach to the molecular analysis methods. So this molecular analysis methods use your DNA barcoding and PCR. This is polymerase chain reaction, and second is of DNA barcoding. This DNA barcoding and this PCR help us to identify the particular specific genes that
Excuse me, ma'am.
will reach us to the, yes,
Ma'am, a few participants are asking to explain in English.
in English. So I'm
Yes, ma'am.
basically, okay, ठीक है. So our next process is morphological analysis and molecular analysis. Morphological analysis, I already told you that when we identify the insects basis on their morphology, like their hair, their formation of wings, their antennas, how their cuticle patterns are formed. These external characteristics. Morphological analysis is basically performed on external characteristics what a particular insect have. Okay. Now, if the examination or differentiation of an insect is not done on a morphological analysis, then we will move to the molecular analysis. Okay, molecular analysis is basically based on nuclear gene sequence identifying. Okay, nuclear gene sequence identifying. For this, we use two methods, PCR and DNA barcoding. PCR require your gene amplification, and DNA barcodings help us to identify that. Yes. Okay. Whatever the questions you have, I will definitely give you about answers after completing the session. So, जो DNA barcoding है, that is DNA barcoding is done for a particular gene of that insect, that is cytochrome oxidase gene. This cytochrome C oxidase gene is very specific to each species. Okay, and it is unique to them only, that it gives the expression that yes, it is belong to that particular family. Okay, DNA barcoding mein what we can do, we have to extract a particular gene on, and extracting a particular gene that is representable, or that is specific or unique to each and every species, that give its structure. Okay, so in COI gene sequencing for a species identification, this is cytochrome C oxidase gene, that is species identification sequencing. It is unique to every species and help us to identify that yes, this species belongs to this family. And how we can identify? Because we already have recorded database. So we can identify that gene to a pre-recorded database and we can compare them. Then next, we will move to the environmental condition. There are various of environmental conditions that are responsible for not forming and forming of the life cycle of a particular insects. Two are basically temperature effects and environmental variations. So temperature effects में first is your accumulated degree hours calculation. Accumulated degree hour calculations means when we are collecting the insects from a kind of crime scenes, then also we have to measure or calculate the particular temperatures or if of environment that is responsible for growth of that particular insects. Okay. Second is a quadratic regression model application. Quadratic regression model application is a parabolic curve that is drawn between the insect life cycle and environmental condition. Okay. Second is SVM. SVM is a species vector model. It is quite complicated. We have to only know this. It is quite complicated. We'll not go deeper in this. So only the thing I will tell you about that. It is a species vector model. It is basically a algorithm, machine learning algorithm. So when we are not clear about the basic characteristics of a particular species, we directly enters the morphological characteristics of that species, like its types of wings, its cuticle formation, its external structures, and so on this. So whatever the data was previously preserved in that SVM database, it will filter the things which we enter in it and give the desirable results. Okay. Second is environmental variables, humidity and precipitation patterns, wind speed and air calculations, seasonal and successional variation, different things like a moist weather and a dry condition and a dark area. These are some environmental conditions that can affect the like uh, decomposition rate of the insects, and also the types of clothes which a decomposed body was having. Maybe if it was in plastic wrap, or if was in normal cloths. They can also affect the rate of formation of the insects. Okay, so these are the different things. Now we reaches to the biological factors that are responsible for the insect colonization. So body factors such as body size, clothing, trauma patterns, and decomposition stages significantly includes insect colonization patterns and development stage. Body size, whether the person was thin or fat, किस तरीके की clothing थी, that like I already told you, that is a normal cloth, that her body was wrapped in a plastic or it was in a like anything else. And the trauma patterns also, like the other different injuries, like it is a open wound injuries or anything that help us to grow the bacteria more. Toxicological effects, the drugs and toxins can alter the larval development rates requiring adjust PMI calculation. Yes, if a person consumed any toxicological poisons or anything, any toxin, then some of the toxins can help us, can help bacteria, can help insects to grow, and some uh, toxins reduce the growth of the insects. Okay. Species factor, individual species have unique temperature thresholds and development requirements, and population dynamics, predator-prey relationships, and competition affect succession patterns. So here are some three or two case studies. First case study is on 24th of May 2022. A 61-year-old male was discovered in an urban residence with extensive maggot colonization. The body was in advanced decay stage with significant insect activity. Temperature data from electronic thermohygrometers combined with Korea Meteorological Administration data enabled precise PMI calculation using established growth models for Lucia and Saligrata. Okay, so what happened in this case, we will tell you ki, the let me just okay. Maggot colonization was found on that particular 61-year-old lady, and the morphological characters was not easy to identify. So in this case, I told you that SVM, that species vector method. In this, the expert what did, he enters the condition what he was observing in the maggots, like whether it is a first insert or second insert or a third insert, then what are its characteristics, and after entering all the data into SVM method, they the correct information about the particular species come out of that. Okay. Second case is was of on August 12, 2022. An 84-old female was found in her living room during advanced decomposition. Third instar larvae were concentrated in facial and neck regions. The body showed characteristic decay patterns with no rainfall recording during the relevant period, providing ideal condition for accurate TMI estimation. So what happened in this? In this stage, the third instar larva was observe onto the decaying body, which help us to identify like how much time the body was taken to decompose. Okay. Now, here is a legal consideration that should be taken in forensic entomology cases to for its admissibility in the courts. So, basically, a forensic entomology evidence serves as a corroborative evidence or a supportive evidences in the courts. It's the expert witness qualification. How much a forensic entomologist is qualified, how much it is data, how much its methods are precise or universally accepted by the other experts also. Okay, it depends what are the publication or books or method he or she has used to examine it. Second is evidence chain of custody. Told you that evidence that collected evidence from the crime scene should be properly documented into the different methods. Already told you no two specimens or no two evidences of the insect should be combined together. Each evidences should be separately packed. So proper documentation, preservation and handling protocols ensure entomological evidence maintains legal admissibility in the court proceedings. And the last is ethical standards and limitations. Professional responsibilities include accurate reporting, acknowledging uncertainties, and avoiding overstatements of conclusions in forensic testimony. Okay. When the expert is giving his testimony, then he or she said that yes, maybe this some of the insects formation may be affected by environmental condition or toxin. Means he or she should be properly known about the statement what he or she going to deliver in the court. Next is a technological advancement. There are different methods from which we can identify the insect species. First is your DNA analysis. I already told you about the DNA barcoding, in which we properly work or examine the mitochondrial cytochrome C oxidase gene that is very specific to every species and give their particular characteristics. Then thermal modeling. Thermal modeling is what we are getting a particular diagrammatic representation of a, body, and where if we see that see this, you can all see that this is a blue color is the area where the is growing, okay, and the green color is the colonization of the particular species. That species is forming into a particular temperature, okay. You know that a particular decomposed body which species form हो रही है that need a particular temperature to grow. So this thermoregulation, this green color is showing by the bacteria that is growing on particular body. Next is the digital documentation. 3D scanning and photogrammetry create detailed crime scene reconstruction. What are the challenges and limitations we faces while examining the body, while examining the PMI estimation? What are some accuracy factors in PMI estimations? So the temperature responsible when there is a low temperature, that low temperature uh, inhibit the growth of the insects and it affects the life cycle. When there is a particular desirable temperature, what exactly the insects require to grow, then it grow in a particular frequency. Species identification also matter, then drug effects also matter. Like some drugs are responsible for retardation the growth of the insects, and some drugs are responsible for a proper growth of the insects. So if we talk about, then there is OPAs that is responsible for the retardation of the insects colonization. And if we take about cocaine, that cocaine is a good drug that responsible for the insect colonization. Environmental factors also responsible, preservation also responsible, and proper documentation is also responsible. Then drug effects on the development alteration. Drugs and toxins significantly alter insects' larval stage, morphology, and behavior patterns. Study shows that cocaine, heroin, ketamine can accelerate or decelerate development by up to 29 hours, requiring adjusted PMI calculation, as I already told you. So, toxicological detection. In some cases, if the body mass is not there, then by identifying the toxical implementation or toxicological changes that are affecting the growth of the insects, we can identify what types of poison is given to the person. Like insects larvae serve as alternative analytes for drug detection when human tissue samples are inedible for or degraded. This capability extends forensic toxicology applications in advanced decomposition cases. Next is your future directions. We can do a research advancement on the future on the forensic entomology, that we can extend the species database and more add the species database, because there is a lots of species is still on the earth that are still not identified or entered their data in the database. We can improve temperature models, we can standardize our protocols, okay, we can enhance more accuracy rates, will help us to identify PMI intervals. Technological integration, we can more added AI assistant identification, real-time monitoring, mobile application, cloud database, and automatic analysis. Training programs should be there. There should be more certificated certification standards, continue education about the forensic entomology. Quality assurance should be maintained by examining the examples, and legal framework. More admissibility of the entomology forensic insects as an evidence should be taken in the course. Evidence protocols should be there, expert qualifications should be there, and more should be added. Quality control should be there, and reporting guidelines, like how should be the insects evidence should be reported or presented into the course. So thank you all. Here's my PPT is ended. Now I'm open for the Q&A.
Thank you, ma'am, for your engaging and
Thank you, ma'am.
informative presentation on forensic entomology. We appreciate your time and expertise you shared.
Thank you so
Participants, if
much.
you have any questions related to the topic, please share your questions in the chat box or ask directly.
Hmm. मैम मॉलिक्यूलर एनालिसिस जब हम करते हैं तो क्या ईच एंड एवरी सैंपल का करना होता है? Yes. Is I'm audible? Hello?
Yes, ma'am.
Is I'm okay. So there is one question that is molecular analysis compulsory for each and every sample? Yes, molecular analysis of each and every sample is very important, because why we are doing this evidence collection? Because we can have that the body is relocated. Okay, maybe body was carried from somewhere from the primary scene and transferred to the secondary crime scene. So in that case, both the crime scenes will have the different types of insects, and maybe they do not similar to each other. So we have to do molecular analysis, because I already told you that in the DNA, the mitochondrial cytochrome C oxidase is the gene responsible for the species identification, and this gene is specific to everyone. So to sorry, every species. So yes, we have to do molecular analysis of each and every sample we are taking from the body, whether we are taking from the any of the body cavity. If we are taking it from the nose, we are taking from the ear, or we are taking from the mouth, or we are taking from the soil sample, we have to analyze it. First, before the molecular analysis, if we can easily identify from the morphological examination, then there is no need for the molecular identification. But if our morphological finding is not clear, then we will go for the molecular analysis. ओके नेक्स्ट क्वेश्चन एज एन आईओ अगर हम एक डेड बॉडी मिला है जो डिकम्पोज हो चुका है पानी के अंदर, लाइक सी और ओशन, और ड्यू टू एनी केस वी डेड बॉडी टेरेस्ट्रियल में आ गया है और रिमेनिंग डिकम्पोजिशन लैंड में हुआ है, तो वाटर एंड लैंड के इंसेक्ट्स को कैसे आइडेंटिफाई करेंगे? सी वाटर एंड हाँ, वाटर एंड लैंड। सी वाटर में आपके डिफरेंट डिफरेंट डायटम्स होंगे बॉडी में जाएंगे, देन डिफरेंट डिफरेंट इंसेक्ट्स जो होंगे वो आपकी बॉडी को इफ ऐसी की बॉडी में डिकम्पोजिशन रेट होगी, बट उन्हें हम डिकम ऐसे तरीके से एग्जामिन करेंगे कि जब बॉडी अगर बाहर सराउंडिंग्स में आपको मिल रही है, तो ऑब्वियसली है कि वो इंसेक्ट्स जो होंगे जो वाटर एरिया एनवायरमेंट में होंगे, वो आपके एनवायरमेंटल कंडीशन में प्रॉपर ग्रो नहीं कर सकते। क्यों? क्योंकि एक तो टेम्परेचर डिपेंडेंट है, जो वाटर में जो आपके इंसेक्ट्स के जो एग्स होंगे या इंसेक्ट्स ग्रो कर रहे होंगे उनका टेम्परेचर वाटर के लिए सेपरेट है, और नॉर्मल एनवायरमेंटल कंडीशन में आने के बाद उनका जो कंडीशन होगी वो सेपरेट होगी, या तो एग्स जो होंगे वो हैच ही नहीं कर पाएंगे। तो उसके बेसिस पे अगर कोई एग को हमने सैंपल में कलेक्ट किया है और देखा है कि उसकी फर्दर प्रोसेस हो ही नहीं पा रही है, क्यों? ड्यू टू द वेरी एनवायरमेंटल कंडीशन एंड वेरी, तो वी कैन एग्जामिन कि ये जो इंसेक्ट्स है ये किस फैमिली से बिलोंग कर रहे हैं। तो इस बेसिस पे हम दोनों को डिफरेंशिएट करेंगे, क्योंकि वाटर कंडीशंस के जो एग्स होंगे या इंसेक्ट्स होंगे वो नॉर्मल एनवायरमेंटल कंडीशन में टेरेस्ट्रियल रीजन्स में प्रॉपर ग्रो नहीं कर पाएंगे। What is the scope of forensic entomology in India? Okay, so it is growing in India. It will take a time, but yes, forensic entomology cases are also growing. Forensic entomologists are also growing in India, but it will take time.
हेलो मैम।
Yes.
मैम क्या क्या चैलेंजेस या लिमिटेशन हो सकता है एंटोमोलॉजिकल एविडेंस को कोर्ट में प्रेजेंट करने के लिए?
Challenges and limitations, see, मैंने आपको पहले ही बताया था. Let me go to the thing. Oh, there's a okay. Challenges और limitations ये हो सकता है कि see, first the thing कि maybe the person should be not well qualified. Okay. the expert who's presenting the evidence, insect as an evidence in the court, he or she should be well qualified. Okay, if we do not have the proper knowledge ki what from how these insects evidence is analyzed and what are the publication, what are the books or the reference material he or she have used to examine it, whether these methods or techniques are universally accepted or not, तो उनके ऊपर क्वेश्चंस हो सकते हैं. ठीक है. देन अगर आपकी चेन ऑफ कस्टडी प्रॉपर फॉलो नहीं की गई है, जो मैंने आपको पहले ही बताया था कि अलग अलग तरीके से आपको अलग अलग सैंपल्स को प्रिजर्व करना होता है. आपको एक ही बॉडी पे मिली डिफरेंट डिफरेंट स्टेजेस को आप एक साथ प्रिजर्व नहीं कर सकते हो. इवन दो आप सेम स्टेजेस को भी एक साथ प्रिजर्व नहीं कर सकते हो अगर आपको वो डिफरेंट डिफरेंट प्लेसेस से मिल रही हैं. आपको उन्हें लेबल करना होगा. अगर आपकी चेन ऑफ कस्टडी डिस्टर्ब हुई है, अगर आपने उनका प्रॉपर कलेक्शन प्रिजर्वेशन नहीं किया है, देन भी आप वो चैलेंज फेस कर सकते हो. देन सेकंड इज एथिकल एंड लीगल इमिटेशन. अगर आप अपने स्टेटमेंट को लेके प्रॉपर श्योर हो और अगर आप प्रॉपर स्टेटमेंट दे सकते हो कि यहाँ डेटा सम अनसर्टेनिटीज फॉर्म ड्यू टू विच वी कैन नॉट प्रॉपर एग्जामिन द पर्टिकुलर इंसेक्ट्स और इंसेक्ट्स को हम प्रॉपर कलेक्ट नहीं कर पाए, यहाँ वहाँ पे जो भी प्रॉब्लम्स आई, तो ये सारी कुछ लिमिटेशंस हो सकती हैं जो कि आपको स्टेटमेंट्स को या लीगल एडमिसिबिलिटी को अफेक्ट कर सकती हैं.
ओके मैम.
Ma'am, shall I read out the questions to you?
Yes.
Aditi is asking, how reliable is entomology when a body is submerged in water, and what insects work on decomposing the body in the water?
Okay, so reliable, if you're talking about, so when a body we are finding in the sea, so basically its decomposition happened, but not that much. ठीक है. एनवायरमेंटल कंडीशंस में आपकी जो आपकी वाटर्स में जो बॉडी मिलती है ना, वो बॉडी आपकी कंप्लीटली ब्लोट हो जाती है ना, वो ज्यादा आपको डिकम्पोज नहीं मिलेगी एज कम्पेयर टू एनवायरमेंटल कंडीशंस. ठीक है. एंड अगर वो सी लेवल मतलब जो बेस लेवल पे आ जाएगी और बेस लेवल पे आके इफ वी फाउंड इट कि अगर वो सी साइड आ गई है या फिर रिवर साइड आ गई है, देन सॉइल कंडीशंस के कांटेक्ट में आके उसमें डिकम्पोजिशन स्टार्ट हो सकता है, बट आपकी वाटर में डिकम्पोजिशन रेट थोड़ी कम हो जाएगी एज कम्पेयर टू दैट सॉइल. एंड इट इज रिलायबल क्योंकि अगर हमें कोई फाइंडिंग्स मिल रही हैं तो हम उसे एग्जामिन कर सकते हैं क्योंकि वी हैव डाटाबेस आल्सो, सो उसके बेसिस पे हम कंपैरिजन कर सकते हैं.
Okay, ma'am. Next question is from Ajay Mandal. Decomposers like fungus and bacteria correlate करते हैं क्या इंसेक्ट्स के डेवलपमेंट में? क्यों डेड बॉडी का कलेक्शन मैनमेड और ह्यूमैनिटेरियन ऑर्गेनाइज्ड भी हो सकता है क्या?
फंगस एंड बैक्टीरियाज. ओके, सी फंगस और आपके बैक्टीरियाज कोरिलेट नहीं करेंगे. फंगस में क्या होंगे कि दैट इज आपके जो बैक्टीरियाज होंगे वो इंटरनली बॉडी को अफेक्ट करना स्टार्ट कर देंगे, ग्रो करना स्टार्ट कर देंगे. ओके. एंड वी कैन मतलब हमें ऐसे फंगस तो नहीं मिलती है डिकम्पोज पे इजीली, बट बैक्टीरियाज देखने को मिल सकते हैं और वो बैक्टीरियाज इंटरनली प्रोलिफरेट करना स्टार्ट होते हैं. क्यों? क्योंकि बॉडी का एक डिफेंस मैकेनिज्म होता है जो उन पे वर्क करना बंद कर देता है जब पर्सन की डेथ हो जाती है. तो वो बैक्टीरियाज जो मैंने पहले ही बताया था वो एनारोबिक बैक्टीरियाज होते हैं जो कि आपकी बॉडी में अगर ऑक्सीजन अवेलेबिलिटी खत्म भी हो जाएगी फिर भी प्रोलिफरेट करेंगे और बॉडी की डिकम्पोजिशन के लिए रिस्पॉन्सिबल होते हैं. ओके, सो ये दोनों सेपरेट पार्ट होंगे. व्हाट वाज द सेकंड क्वेश्चन? ह्यूमैनिटेरियन समथिंग लाइक?
क्यों डेड बॉडी का कलेक्शन मैनमेड और ह्यूमैनिटेरियन ऑर्गेनाइज्ड भी हो सकता है?
मैनमेड और ह्यूमैनिटेरियन, कैन यू एलैबोरेट दिस?
Ajay Mandal, please ask your question. ऑलराइट, मे बी शायद ही इज आस्किंग इंसेक्ट्स के डेवलपमेंट में डेड बॉडी का कलेक्शन मैनमेड और ह्यूमैनिटेरियन ऑर्गेनाइज्ड हो सकता है क्या?
डेड बॉडी पे अगर इंसेक्ट्स फॉर्म हो रहे हैं तो कलेक्शन ऑफ इंसेक्ट्स कैन बी मैन मेड.
मे बी, आई डोंट.
No, I'm also not getting this question. What he is exactly about to ask.
Ajay Mandal, if you are there, please ask your question directly.
He is also not getting what he is asking.
गुड मॉर्निंग मैम.
Yes, good morning.
मैम मेरा क्वेश्चन था कि जैसे कि अगर हम लोग डेड बॉडी हमको अगर हमको कलेक्शन हुआ है तो मैम जैसे कि बहुत
हाँ
से जो ह्यूमैनिटेरियन भी चेंज कर सकते हैं.
हाँ
मतलब वो कहीं और उनका प्राइमरी कलेक्शन रेट कहीं और रहता है लेकिन वो मिलता कहीं और पे है. तो मैम उसमें
हाँ
क्या मतलब इंसेक्ट का जैसे जो इन्वायरमेंटल जिससे होते हैं ना कि मैम जैसे कि
हाँ
अगर बॉडी कोई गर्म इलाका में मिल रहा है वहाँ पे इंसेक्ट का अलग वो होता है और ठंडा
हाँ
इलाके में अलग होते हैं. तो मैम
हाँ.
उसमें कोई चेंजेस या डेवलपमेंट का ऐसे कोई इम्पैक्ट मतलब इम्पैक्ट होता है क्या?
अच्छा, ओके. मतलब आप ये बोल रहे हैं कि जो आपका बॉडी जो है वो अगर डिफरेंट डिफरेंट एरियाज में मिल रही है,
जी
जैसे
मैम.
कि कूलर
यस मैम.
एरियाज और हॉट ह्यूमिड एरियाज, तो उसमें कैसे तरीके का कलेक्शन होगा ऐसे?
यस मैम. मतलब जैसे होता है कि मतलब अगर वो जो प्राइमरी जो डेड बॉडी मिला है वो
हाँ हाँ
कहीं
हाँ
और था तो वो
हाँ
इंसान
प्राइम.
मतलब जो क्रिमिनल होता है उसको कहीं
हम्म.
और फेंक दिया तो मैं उसके
हाँ
लिए.
हाँ 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 सम ओके ओके, गॉट इट गॉट इट. देखो, बेसिकली क्या होता है कि मैंने आपको पहले ही बोला था कि जब आप एविडेंस कलेक्शन कर रहे हो तो एविडेंस कलेक्शन में आपका जो मेन मोटिव जो है वो यही है कि कहीं बॉडी को रिलोकेट तो नहीं किया गया है. ठीक है. तो
यस
अगर
मैम.
आपका बॉडी प्राइमरी क्राइम सीन पे है जहां पर आपका टेम्परेचर जो है बहुत लो है, ठीक है, और सेकेंडली बॉडी को आपका टेम्परेचर बॉडी को ऐसी जगह पे शिफ्ट कर दिया गया है जहाँ पे कि बॉडी का टेम्परेचर या एनवायरमेंटल कंडीशंस आपकी ह्यूमिड है, ड्राई है, हॉट एनवायरमेंट
जी
है,
मैम.
तो ऐसे केसेस में वो क्या होगा कि पहला वाला केस हमने लिया जैसे कि बॉडी बहुत कोल्ड एरिया में थी, ठीक है. तो सबसे पहले आपको बता दूं कि कोल्ड एरियाज में अगर बॉडी होती है ना तो वहां पे बैक्टीरियल का ग्रोथ भी कम होता है. हम बॉडी को क्यों मॉइस्ट में रखते हैं कूलर टेम्परेचर में? ताकि बॉडी में जो है बैक्टीरियल ग्रोथ रिटार्ड हो जाए, या उसकी प्रोसेस अगर हो तो बहुत स्लो हो या बहुत देर से हो. तो जब बैक्टीरियल प्रोलिफरेशन नहीं हो रहा है मतलब बॉडी डिकम्पोज नहीं हो रही है. बॉडी डिकम्पोज नहीं हो रही है इसका मतलब कि बॉडी में वो जो गैस फॉर्मेशन हो रहा है वो भी नहीं हो रहा है. वो गैस फॉर्मेशन नहीं हो रहा है मतलब कि वो फॉल ऑर्डर स्मेल नहीं जा रही है. फॉल ऑर्डर स्मेल नहीं जा रही है मतलब वो किसी भी तरीके की फ्लाइज या क्या जो आपके इंसेक्ट्स है उसको अट्रैक्ट ही नहीं कर रही है. तो ये स्टेज पहले आपकी स्टार्ट ही नहीं होगी. क्लियर? दूसरा, अगर बॉडी को आप दूसरे जगह पे लाके ट्रांसफर कर देते हो तो अब आपकी ये चेंजेस आ सकते हैं कि धीरे धीरे बॉडी का टेम्परेचर कम होगा और फिर वो एक्सटर्नल एनवायरनमेंट के अकॉर्डिंग सेटल होगी. फिर वो बॉडी में वो डिफरेंट डिफरेंट बैक्टीरियल ग्रोथ स्टार्ट होंगी. और अगर इफ इन केस बॉडी पहले कोई ऐसे कोल्ड एरिया में थी भी जहाँ पे अगर डिकम्पोजिशन हो भी गया था और वो इंसेक्ट्स थे भी, तो अगर आप बॉडी की प्लेस को चेंज करोगे और अगर आप उसको नॉर्मल एनवायरमेंटल कंडीशन में लेकर आओगे तो वो बैक्टीरिया और वो इंसेक्ट्स जो पहले आपकी ग्रो कर पा रहे थे उस कोल्ड एनवायरमेंट में, वो आपके नॉर्मल एनवायरमेंटल कंडीशन में ग्रो नहीं करेंगे, उनकी ग्रोथ रिटार्ड हो जाएगी. और अगर हमें वो बॉडी पे मिलेंगे भी और अगर हम उन्हें कलेक्ट करेंगे भी तो हम क्यों एग्स को और हम इंसेक्ट्स को और हम लार्वा को इस तरीके से कलेक्ट कर रहे थे कि एक तो उसको हम किल करके प्रिजर्व कर रहे हैं और एक हमको एज इट इज कलेक्ट कर रहे हैं. क्यों? क्योंकि हम देखना चाह रहे हैं कि जो किल कर दिया है वो वैसे के वैसे ही स्टेज पे है और जो ग्रो कर रहे हैं तो जो हम उसे ग्रो कर रहे हैं तो वो जो इंसेक्ट्स एडल्ट स्टेज में आ रहे हैं तो उनका मॉर्फोलॉजिकल स्टेजेस उससे मैच कर रहा है कि नहीं, जो हमने रिसेंटली नॉर्मल एनवायरमेंटल कंडीशन से जो कलेक्ट किए हैं बैक्टीरिया सॉरी इंसेक्ट्स उनसे. तो इस तरीके से वेरी करेगा. इंसेक्ट्स आपके टेम्परेचर डिपेंडेंट होते हैं. जो इंसेक्ट्स आपके टेम्परेचर में ग्रो कर पा रहे हैं वो दूसरे टेम्परेचर में ग्रो नहीं कर पाएंगे. इस तरह से हम इजीली आइडेंटिफाई कर सकते हैं उनकी लाइफ साइकिल्स को आइडेंटिफाई करके.
वही उसी चीज पे डाउट था कि जैसे मैम बहुत बहुत सारे केसेस ऐसे भी होते हैं कि मैम
हम्म
जैसे उनका जो मर्डर हुआ है वो कोल्ड एरिया में हुआ है लाइक शिमला और
हम्म
समथिंग. उसके बाद उनको
हम्म
जानबूझ के ताकि उनका जो डेथ का टाइम हो उसको मैनिपुलेट करने के लिए उनको कहीं ऐसा
मैनिपुलेट.
एरिया में रख दिया जाता है. हाँ ऐसा
हाँ हाँ
एरिया में
हाँ
रख दिया जाता है तो परफेक्ट जो डेट ऑफ टाइम होता है ना वो पता ना कर पाए.
देखो एक तो
इसलिए
एक
मैम।
ये चीज भी, हाँ ये चीज भी अफेक्ट करती है कि जब आप जैसे टेम्परेचर चेंज कर, देखो जो बॉडी के पोस्टमॉर्टम इंटरवल्स होते हैं ना, बहुत सारे इंटरवल्स होते हैं। जैसे कि आपका फ्रेश डिके होगा, पहले बॉडी में रिजिडनेस आएगी, फिर बॉडी में एल्गर मोटिस, लाइवर मोटिस